DNA. It's basically the ultimate director for our cells. It codes for everything that makes us, well, us. But with a job that important, what happens when a cell needs to divide? Simple. It needs a perfect copy of those instructions. And that process is called DNA replication. Today, we're going to unpack that blueprint for life. So that brings us to the big question, right? How does one single cell with its one complete set of instructions become two cells where each one has its own perfect copy? The answer is hiding in this elegant and just incredibly precise molecular process that happens before every single cell division. The best way to think about DNA replication is like a super specialized construction project. And, every project needs a crew. So let's meet the key players. Most of them are enzymes. They're the biological workers that make reactions happen fast. A little tip-off in biology, you'll notice a lot of them end in ase, which is a pretty good sign you're dealing with an enzyme. Okay, first one on the scene is helicase. This is our unzipping enzyme. Its whole job is to tackle that famous double helix. It just cruises along the DNA and breaks the bonds holding the two strands together, effectively unzipping the whole molecule to get at the genetic code inside. Next up, we've got the real star of the show, DNA polymerase. This is the master builder. This enzyme's job is absolutely critical. It actually builds the new strand of DNA. It reads the original strand and then grabs the correct complementary bases, A with T and G with C, and just constructs a brand new, perfectly matched partner strand. But here's the thing. As amazing as DNA polymerase is, it can't just start building wherever it wants. It needs a signal, kind of like a starting block for a race. And that's where primase, the initializer, comes in. It makes this short little piece of RNA, called a primer, that basically tells DNA polymerase, okay, start building right here. And finally, in our main crew, we've got ligase, the gluer. Now, as we're about to see, sometimes the new DNA gets built in little fragments instead of one long piece. Ligase is the finishing specialist that comes in and seals up all the gaps, making sure the new strand is a single, continuous, and solid piece of DNA. So we've met the crew. How did they get started? Well, just like any good construction project, before any building can happen, they've got to prepare the worksite. First things first, the crew has to find the starting point, which is called the origin. Then helicase gets to work on zipping. But, a couple of problems pop up right away. Problem one, those single strands want to snap right back together. The solution? These little things called single-stranded binding proteins, or SSB proteins. They jump on and hold them open. Problem two, as you unwind the DNA, the part up ahead gets all tangled up in supercoils. So another enzyme, topoisomerase, comes in to relieve that tension and keep the track nice and clear. All right, now this is where things get really, really interesting. The replication crew runs into a major challenge. Because of one simple, fundamental rule, they're forced to use two completely different building strategies at the exact same time. So what's the deal? Why is building new DNA kind of like driving down a one-way street? It all comes down to a concept called directionality. See, the two strands of DNA are antiparallel. They run in opposite directions. And that little fact changes everything. Okay, so here's the crucial rule that creates this whole puzzle. Our master builder, DNA polymerase, can only add new bases to one specific end of a growing DNA strand. In molecular terms, we say it can only build in the 5' to 3' direction. This single rule dictates the entire construction process. So because of that one-way rule, one of the new strands is super easy to build. It's called the leading strand, and it gets built smoothly, continuously, in one long piece. It just follows right along behind the unzipping helicase. Easy peasy. But the other strand, the lagging strand, well, that's a whole different story. It's pointing in the wrong direction, so it has to be built backwards, discontinuously, in all these small, stitched-together pieces called Okazaki fragments. Yeah, this lagging strand is where things get really complex. It's not one smooth job at all. Instead, primase has to keep laying down new primers, one for each little fragment. Then DNA polymerase builds backwards in short bursts. And finally, our gluer, ligase, has to come in and stitch all these separate Okazaki fragments together to make one complete strand. It's a much more complicated process. So the crew has built two new strands of DNA. The main construction is done, but the job isn't over. Not by a long shot. Now it's time for some absolutely critical quality control to make sure the job was done perfectly. And the final product? It's just incredible. From that one original double helix, the cell now has two absolutely identical DNA molecules. Each one is a perfect blueprint for life, ready to be passed on to a new daughter cell. You know, there's a cool scientific name for this process: semi-conservative replication. And it's called that because it half conserves the original. Each of the two new DNA molecules has one of the original, old strands and one brand new strand. This is a brilliant system that makes sure the original template is perfectly preserved and passed on in every single copy. But you might be wondering, how accurate is all this? I mean, a mistake could be a disaster, right? Well, get this. DNA polymerase isn't just a builder, it's also a fantastic proofreader. It checks its own work as it goes, and its accuracy is just staggering. It makes only about one mistake for every billion bases it adds. 1 in a billion. That is an unbelievable level of precision. Okay, so we've gone deep into the how it's done, but why? Why does this tiny, microscopic dance of enzymes matter so much in the grand scheme of things? Well, this process is, and I'm not exaggerating, the foundation for almost everything in biology. It's essential for cell division, which lets us grow and repair our tissues. It's how genetic information gets passed on. And beyond that, understanding it has led to some of our most powerful medical treatments that can actually stop replication in harmful cells, like bacteria or even cancer. And that brings us to a final, pretty powerful thought. This process is the engine of life, for sure, but it can also be the engine of disease. Now that we understand the blueprint, the really big question becomes, what if we could control this process with even greater precision? What if we could stop harmful cells from ever replicating again?